Venezuelan Ancestry. Venezuelan ancestry spans a wide mix of indigenous, European, and African roots, but along the shores of Lake Maracaibo in Zulia, a more concentrated history took shape. In isolated fishing villages, large extended families grew over generations, creating one of the most striking examples of how geography and lineage can quietly amplify a single genetic variant. Here, Huntington's disease appears at an unusually high frequency. It's caused by an expanded repeat in the HTT gene. Over time, it gradually damages the brain circuits that control movement, mood, and thinking. Early signs can be subtle: restlessness, mood changes, but as the disease progresses, involuntary movements, memory loss, and loss of independence become unavoidable, turning ordinary motion into effort and then dependence. This is not an inherited advantage, but a founder effect, one ancestral mutation carried forward through tightly connected communities, where location itself becomes a risk. The burden extends beyond biology. Because Huntington's is autosomal dominant, each child of an affected parent faces a 1 in 2 chance of inheriting the condition, shaping family decisions, silence, and stigma across generations. In diaspora settings, that history can be obscured, delaying recognition until symptoms have already begun. In this case, risk is written less in ancestry than in place in lineage. The mutation doesn't disappear, it waits and then returns. Colombian Ancestry. Colombian ancestry spans a wide geographic and genetic range, from Andean highlands to Caribbean coasts, shaped by indigenous foundations, Spanish colonization, and African diaspora. These layers did not blend evenly. Instead, they formed regional pockets, where certain traits, protective or harmful, could become concentrated within families over generations. In Antioquia, a large extended family network carries an inherited mutation in the PSEN1 gene that causes early-onset Alzheimer's disease. Symptoms can begin decades earlier than expected, with memory loss and cognitive decline appearing in what should be midlife. The mutation disrupts how proteins are handled in the brain, speeding degeneration far earlier than in typical Alzheimer's. Over generations, this mutation became concentrated within interconnected families, affecting entire family lines rather than isolated individuals. As a result, this region has become one of the most closely studied Alzheimer's populations in the world. Certain BRCA1 and BRCA2 mutations also appear more frequently in some Colombian lineages, where impaired DNA repair raises the risk of breast and ovarian cancers. Broader admixture adds another layer. In some regions, African ancestry contributes to variants linked to kidney disease risk, while indigenous ancestry can influence lipid metabolism, increasing susceptibility to fatty liver as diets shift. These influences do not act uniformly. They depend on where families come from and how their environments have changed. What appears to be chance begins to look predictable once the family line is fully traced. Navajo Ancestry. The Navajo trace their ancestry to Athabascan-speaking peoples who settled the high, arid plateaus of the American Southwest, adapting to scarce water, wide distances, and a demanding climate. Their history includes forced displacement during the Long Walk and later exposure to mining economies, leaving communities both geographically isolated and shaped by population bottlenecks that can amplify rare inherited conditions. Two inherited disorders stand out because of how consistently they appear within certain families. Artemis severe combined immunodeficiency, caused by a genetic mutation, prevents the immune system from developing properly, leaving infants without the T-cell defenses needed to fight even routine infections. Navajo neurohepatopathy, also linked to mutations in the MPV17 gene, disrupts mitochondrial DNA maintenance, impairing energy production in the liver and nervous system and leading to severe, often early life illness. These conditions persist not because they offer any advantage, but because of shared ancestry within relatively isolated communities, where rare variants can be passed down and become more visible over time. A different layer of risk comes from the environment. Legacy uranium and arsenic exposure from mining can place chronic stress on kidneys and development, especially when combined with dehydration and limited access to healthcare in a dry climate. Here, vulnerability reflects both inheritance and exposure, where long-standing genetic patterns intersect with more recent environmental pressures. Cuban Ancestry. Cuban ancestry grew out of the layered history of the island itself, Spanish colonization, African diaspora, and the surviving imprint of indigenous Caribbean ancestry. That history did not leave a single medical signature. Instead, it produced a mosaic, with some risks spread broadly and others concentrated so tightly in particular regions and families that they remain invisible until symptoms begin. In eastern Cuba, especially around Holguin, certain family lines carry spinocerebellar ataxia type 2. Caused by an expanded repeat in the ataxia gene, it gradually breaks down the systems that control balance, eye movement, speech, and coordination. Movements that once felt automatic become unstable. Walking requires effort. Speech slows. Over time, even basic coordination becomes difficult to maintain. This pattern reflects how a rare mutation can become concentrated within a specific region and its family networks, where it is passed down across generations. A different risk can appear through inherited European ancestry. In HFE-related iron overload, the body absorbs more iron than it needs, often for years without symptoms, until damage begins to affect the liver, heart, or pancreas. Some risks are quieter, but still present. Hemoglobin variants remain clinically relevant in certain families, while migration can obscure these patterns when medical histories are reduced to broad ethnic labels without attention to regional origin. Some conditions remain hidden until they surface within the same families, in the same places, generation after generation. Puerto Rican Ancestry. Puerto Rican Ancestry formed at the crossroads of empire and survival, where indigenous Caribbean communities, Spanish colonists, and enslaved Africans were brought together on a humid, storm-battered island. In towns that often remained locally rooted, this tri-continental mixture created both rich genetic diversity and tightly clustered lineages, allowing rare variants carried by a few individuals to expand quietly over generations. One example is Hermansky-Pudlak syndrome, which appears at unusually high frequency in certain regions because early carriers pass the mutation through closely connected communities. It affects structures inside cells that act like recycling centers, leading to reduced pigmentation, fragile blood clotting, and in some forms, progressive lung scarring that gradually makes breathing more difficult. A separate risk appears in a Puerto Rican founder mutation in BRCA2, a gene responsible for repairing damaged DNA. When that repair system is impaired, errors accumulate, increasing lifetime risk of breast and ovarian cancers. Some risks are less concentrated, but still important. Asthma appears at high rates, shaped by immune sensitivity interacting with mold, pollution, and viral exposures. Diabetes reflects a similar interaction, where metabolic traits meet modern diets, rich in processed foods, and more sedentary routines. Even diagnosis can fall short when simplified racial categories overlook this layered ancestry. Puerto Rican ancestry reflects both diversity and concentration, where widely shared risks and highly localized ones exist side by side. What matters most is not just ancestry itself, but how precisely it is understood. Brazilian Pardo Ancestry. Brazilian Pardo Ancestry emerged from centuries of forced migration, colonization, indigenous continuity, and later settlement, bringing together Portuguese, African, Native American, and sometimes Japanese lineages into one of the most layered genetic landscapes in the Americas. Across rainforests, coasts, plantations, and inland frontiers, traits shaped by malaria, famine, and regional isolation were carried forward into a very different medical environment. Some of the clearest examples appear in hemoglobin disorders. Sickle cell trait persisted because carrying a single copy offered protection against severe malaria, while two copies can lead to sickle cell disease, where red blood cells stiffen under low oxygen, damaging blood vessels, lungs, and the brain. Thalassemias follow a related pattern. Reduced hemoglobin production may have once offered an advantage, but today can result in chronic anemia, repeated transfusions, and iron overload. In some Japanese Brazilian lineages, ALDH2 variants slow the breakdown of acetaldehyde, making alcohol both immediately uncomfortable and, over time, more carcinogenic. Another inherited condition, G6PD deficiency, leaves red blood cells more vulnerable to oxidative stress, which can be triggered by infections, certain medications, or even specific foods like fava beans. Brazilian Pardo ancestry reflects the layering of multiple evolutionary pressures, where traits shaped in very different environments now coexist within the same population. What once provided protection in one setting can create vulnerability in another. Barbadian Ancestry. Barbadian Ancestry was shaped on a small Caribbean island marked by British colonial rule, plantation labor, and the forced migration of West Africans. In that confined setting, where communities grew from relatively small lineages and later spread through migration, older pathogen pressures now intersect with modern chronic disease in ways that are easy to miss. A key part of that story is APOL1. These kidney risk variants were inherited from African ancestors because they provided protection against deadly trypanosome infections. But in the present, Carrying two high-risk copies can turn that earlier advantage into a long-term vulnerability. The cells responsible for filtering blood in the kidneys become more susceptible to damage, increasing the likelihood of scarring diseases and, over time, chronic kidney failure. That risk can be intensified by other factors. Hypertension, which is common across the Caribbean, places constant pressure on kidney blood vessels. When it overlaps with APOL1-related risk, the progression toward kidney damage can accelerate. Diagnosis can also be complicated. Broad racial labels in U.S. healthcare settings may obscure Caribbean-specific ancestry, delaying the kind of screening or counseling that could identify risk earlier. Barbadian ancestry reflects a clear evolutionary trade-off. What once helped survival can carry long-term consequences in a different environment. Mexican American Ancestry. Mexican American ancestry often traces to the meeting of indigenous Mesoamerican societies and Spanish colonization, shaped over centuries of farming cycles, periodic scarcity, and maize-centered diets, rather than dairy pastoralism. In these environments, energy efficiency and resilience to fluctuating food supply were valuable, shaped under infectious pressure and physically demanding living. A well-studied example is a type 2 diabetes-associated variant, which changes how cells handle lipids, increasing metabolic strain when calories are abundant. In earlier conditions, this shift may have been neutral or even useful, but in a setting of constant sugar and processed food, it can push the body toward insulin resistance. Changes in cholesterol handling add another layer. Variants can reduce the formation of protective HDL particles, contributing to lower HDL levels and higher metabolic risk, even though they may have been favored in earlier populations. Liver metabolism can also be affected. A genetic variant increases susceptibility to fatty liver disease, especially as diets become more calorie-dense. Mexican American ancestry highlights that small differences in how cells process nutrients can lead to much larger effects across the body. Peruvian Ancestry. Peruvian Mestizo ancestry reflects a long history of life across extremes. Indigenous Andean populations adapted to thin air at high altitude, later blending with Spanish settlers across mountains, deserts, and forests. In the Andes, survival meant extracting oxygen from scarce air, sustaining pregnancies in low oxygen conditions, and maintaining energy balance in harsh, variable environments. One result of that history appears in chronic mountain sickness. At high elevations, the body produces more red blood cells to carry oxygen, an essential adjustment in thin air. In some individuals, however, this response overshoots, thickening the blood and placing strain on the heart and lungs, leading to fatigue, headaches, and increased risk of stroke. Pregnancy adds another layer. While Andean adaptations can improve blood flow to the placenta, altitude still raises the risk of complications, especially when modern stressors disrupt those protective patterns. A different kind of risk emerges outside the mountains. A PNPLA3 variant, found at high frequency in some Peruvian populations, changes how the liver processes fat, increasing susceptibility to fatty liver disease as diets become more calorie-dense. Urban migration can intensify this shift, as metabolic systems adapted for scarcity meet constant abundance. Peruvian ancestry reflects adaptations that help survival at altitude can create unexpected risks in different environments. Mayan Ancestry. Mayan ancestry spans a constellation of indigenous peoples across Central America, shaped by long histories of agriculture, cultural continuity, and communities that often remained locally rooted across generations. In many regions, that continuity created tightly connected family networks where both traditions and rare genetic variants could persist over time. In one documented case, a deletion in the GJB2 gene, essential for inner ear function, led to congenital hearing loss within a Mayan family. When this gene is disrupted, sound signals cannot be properly transmitted, and hearing loss is present from birth. Conditions like this do not spread broadly across populations. Instead, they can remain concentrated within specific families, only becoming visible when two carriers pass the variant on to a child. This reflects how small community structure and endogamy can shape inheritance. Variants that are rare globally can persist quietly within a local group, carried across generations without being recognized. Other health patterns are shaped more by changing conditions than single genes. As diets and lifestyles shift, diabetes and hypertension have increased in some Maya populations, while lactase non-persistence can make dairy-heavy diets difficult to tolerate. In Maya communities, risk often emerges within families rather than across entire populations. What is rare globally can become familiar within a single lineage. Mapuche Ancestry. Mapuche Ancestry is rooted in the temperate forests and coastal regions of southern Chile and Argentina, where communities lived for generations, with distinct food systems, mobility patterns, and social structures. Despite colonization and displacement, many Mapuche lineages remained regionally continuous, shaping patterns of health that are now visible in modern populations. A well-documented pattern is an elevated risk of gallbladder cancer in individuals with higher proportions of Mapuche ancestry in Chile. This risk often begins with gallstones, hardened deposits that form when bile becomes oversaturated with cholesterol. In many Mapuche-descended groups, gallstones are unusually common, increasing the likelihood of chronic inflammation in the gallbladder. Over time, that repeated irritation can lead to cellular changes that raise the risk of cancer, often detected late. Several factors can push this process further. Diets higher in processed foods can increase cholesterol imbalance and stone formation, while limited access to early surgical care may delay removal of high-risk gallbladders. In diaspora settings, this pattern can also be missed when ancestry is reduced to broad categories like Latino. Mapuche Ancestry highlights how risk can develop through a sequence of small steps rather than a single cause. The danger is not immediate. It builds up quietly, one step at a time. Ojibwe Ancestry. Ojibwe Ancestry is rooted in the Great Lakes region, where communities lived for generations in forested environments shaped by long winters, seasonal food systems, and strong local continuity. In some communities that remained relatively small and closely connected, rare inherited conditions became more visible over time. In related Ojibwe populations, certain neurological disorders appear at higher frequency than expected. These include conditions that affect early development, where children may experience delays in movement, learning, or coordination. Some are leukodystrophies, where the protective covering around nerves gradually breaks down, impairing walking, speech, vision, and cognition over time. While each condition is rare on its own, their visibility increases in settings where the same variants are carried within connected family networks. In some cases, specific inherited disorders, such as Fabry disease, have been identified, where early symptoms like burning pain in the hands and feet can later progress to kidney, heart, or neurological complications. This pattern does not reflect a single mutation or a single disease. Instead, it reflects how rarity can cluster and become recognizable within a community. Ojibwe Ancestry highlights how population structure shapes what becomes visible. Rarity changes meaning when it is shared within the same community. Cherokee Ancestry. Cherokee Ancestry is rooted in the southeastern United States, shaped by agricultural lifeways and later disrupted by forced removal along the Trail of Tears. In the generations that followed, communities rebuilt across Oklahoma and beyond, carrying both continuity and the effects of rapid environmental and social change. One of the clearest patterns is how early type 2 diabetes can begin. When elevated blood sugar appears in adolescence or young adulthood, the body is exposed to decades of metabolic strain. Over time, this increases the likelihood of complications such as kidney damage, nerve injury, and vision loss much earlier in life than expected. This is not driven by a single gene, but by how metabolic systems respond to changing diets, reduced physical activity, and long-term stress. Gallbladder disease adds another layer. Gallstones are more common in many indigenous populations, particularly among women, and can lead to inflammation or, in rare cases, more serious complications. A less visible challenge comes from how data is organized. In many healthcare systems, Cherokee individuals are grouped into broad American Indian, Alaska Native categories. That can obscure community-specific patterns and make it harder to track which risks and which interventions apply locally. Cherokee Ancestry highlights how timing and classification both matter. When risk starts earlier but remains poorly defined, it becomes harder to stop. Yanomami Ancestry. Yanomami Ancestry is rooted in the Amazon rainforest along the Brazil-Venezuela border, where semi-isolated villages sustained life through forest-based diets, low salt intake, and minimal exposure to many outside pathogens. For most of their history, health risks were shaped by that environment. In recent decades, however, contact with outside populations, industries, and medical systems has introduced entirely new pressures. Viral hepatitis is one of the most serious. Studies in Yanomami communities have found widespread markers of hepatitis B and D infection, reflecting transmission in settings where vaccination and consistent care are difficult to maintain. These infections can progress quietly over years, eventually leading to liver scarring, cirrhosis, and cancer. Environmental exposure adds another layer. Mercury used in illegal gold mining contaminates rivers and food sources, and testing has found widespread exposure in Yanomami villages. As a neurotoxin, methylmercury can impair brain development, especially in children. Some risks only appear when modern treatment is introduced. Variants in multiple genes affect how certain medications are processed, and reduced activity can cause toxic buildup, leading to severe bone marrow suppression if standard doses are used. Yanomami Ancestry highlights a different kind of vulnerability. In this case, exposure, not inheritance, becomes the defining risk. Dominican Ancestry. Dominican Ancestry reflects a Caribbean blend of Spanish, West African, and indigenous Taino roots, with many families now living in diaspora communities, such as New York City. In clinical settings, this ancestry can be misunderstood, particularly when broad labels like Latino are used without considering underlying genetic variation. One important example is sickle cell trait. In some Dominican populations, carrier rates are high enough that two individuals without symptoms can still have a child with sickle cell disease. Because hemoglobin disorders are not always expected in Latino patients, screening may be delayed or overlooked, allowing risk to go unrecognized until complications appear. At the same time, many Dominican families are affected by high and rising rates of type 2 diabetes. When combined with hypertension, this increases the likelihood of kidney and cardiovascular disease over time, particularly when access to consistent care is limited. These patterns do not act independently. When inherited risk, expectations in clinical practice, and barriers to care intersect, conditions that are manageable can become more difficult to control. Dominican Ancestry highlights how assumptions shape outcomes. What is overlooked is often what causes the most harm. Indo-Caribbean Ancestry. Indo-Caribbean Ancestry traces to South Asian indentured laborers brought to Guyana, Trinidad, and neighboring regions after emancipation, later blending with Caribbean environments and communities. Across generations, this history carried forward a metabolic profile shaped under very different nutritional conditions than those found today in diaspora settings. A key pattern is a form of diabetes risk that can remain hidden in plain sight. Individuals may appear relatively lean, yet carry higher levels of fat around internal organs, especially in the liver, which drives insulin resistance and earlier onset of type 2 diabetes. Because outward appearance does not always reflect this risk, it can go unrecognized until complications begin. Heart disease often follows a similar path. Imbalances in cholesterol, combined with long-term metabolic strain, can accelerate the buildup of plaque in arteries, increasing the likelihood of heart attacks at younger ages than expected. Additional inherited factors add complexity. Thalassemia trait, more common in South Asian lineages, is usually silent but becomes important in family planning when two carriers are involved. Medication response can also vary, meaning standard doses may not always match how the body processes certain drugs. In this ancestry, risk is often not visible on the surface. What appears healthy can still carry underlying strain that builds over time. French-Canadian Ancestry. French-Canadian Ancestry, particularly in Quebec's Charlevoix and Saguenay-Lac-Saint-Jean regions, traces back to a remarkably small founding population of 17th century settlers. Geographic isolation, large family sizes, and centuries of limited migration allowed chance mutations to spread widely, creating one of the clearest examples of founder effects in human genetics. Several rare disorders reach unusually high frequencies. RSACs, a progressive neurological condition, emerges in early childhood with muscle stiffness, balance loss, and impaired coordination. Hereditary tyrosinemia type 1 reflects a metabolic failure to break down an amino acid, once causing fatal liver disease in infancy before modern treatments intervened. Other conditions, such as Anderman syndrome, disrupt both brain structure and peripheral nerves, while vitamin D-dependent rickets weakens growing bones despite adequate nutrition. Even more familiar diseases appear in distinct forms. Cystic fibrosis is common, but driven by Quebec-specific mutations rather than the variants seen elsewhere in Europe. None of these traits offered clear evolutionary advantage. They spread through isolation, not selection. French-Canadian genetics reveal how history can concentrate risk. A small beginning, echoed across generations, turned rare mutations into regional legacies. Quiet reminders that ancestry can shape health in highly local, deeply personal ways. Métis Ancestry. Métis Ancestry formed through the meeting of First Nations communities and European settlers across North America, creating distinct populations shaped by mobility, trade networks, and later displacement. Today, one of the less visible challenges is not only biological, it's informational. Compared with other populations, Métis communities are often underrepresented in large genetic and medical data sets. That gap can affect how risk is recognized and how treatment decisions are made. In pharmacogenetics, for example, differences in how individuals process certain medications may not be fully captured, making standard dosing less precise or harder to interpret. Health patterns still emerge, but they are often studied with less resolution. Rates of type 2 diabetes and cardiovascular disease are elevated in many contexts, driven by a combination of metabolic stress, changing diets, and unequal access to preventive care. These conditions tend to cluster together, increasing long-term strain on the heart, blood vessels, and metabolic systems. Even dietary guidance can be inconsistent. Mixed ancestry means lactase persistence varies, so recommendations built around high dairy intake may not work well for everyone. Métis Ancestry highlights a different kind of disadvantage, one where gaps in data and representation can shape outcomes alongside biology. Here, the absence of data can become a risk in itself. If you like this video and want to learn more about different genetic ancestries, check out these other videos.